What I'm referring to here is the polymerase chain reaction, which is more well known by the acronym PCR. Now, the reason that so many people are familiar with that acronym PCR is because in recent years, millions of PCR tests have been performed worldwide to determine whether individuals have been infected with a particular virus. You know the virus I'm referring to. However, PCR has much broader application than just detecting a virus. Generally speaking, PCR is a technique whereby researchers can amplify many copies of a particular DNA sequence. And when I say many copies, I mean millions or billions of copies of a sequence. And when you have lots and lots of copies of a DNA sequence, such as with PCR, it makes it much easier to determine the presence or absence of a particular sequence. So how can this be used to distinguish between males and females? Well, I first need you to consider two different genes. One of them is found on the X chromosome and one of them is found on the Y chromosome. Now, these genes are very similar to each other and they are referred to as the amelogenin gene. Now, the amelogenin gene is actually a gene associated with tooth enamel. However, what the gene encodes is not really that important. What is important is that the amelogenin gene found on the X chromosome is a little bit different from that on the Y chromosome. It's actually 6 DNA letters shorter. This is how one professor of genetics describes the situation. The amelogenin gene, which produces a tooth enamel protein, is present on both X and Y chromosomes, but the X chromosome version is 6 bases shorter, and this difference can be detected by PCR analysis. When a DNA sample from an unknown donor or suspect is tested for the amelogenin locus, the results for a male who carries both Y and X chromosomes shows 2 separated bands or peaks, and just 1 band for females with only X chromosomes. So you see, if your PCR tests manage to amplify both versions of the amelogenin gene, the version on the X chromosome and the version on the Y chromosome, you obviously have an individual who's genetically male, XY. Alternatively, if your PCR tests only manage to amplify the version of the amelogenin gene that's found on the X chromosome, you obviously have an individual who's female because no Y chromosome is present.